So we have seen the structure of HIV. Next, now we are going to discuss the transmission. How this HIV they can transfer from one person to another person. So HIV is often located within the cells, especially in the macrophages. So where it is located in the macrophages. So monocyte, it is involving in the immune response, they can produce the macrophages. So this HIV they can go on located inside the macrophages. Okay, so HIV can survive for 1.5 to inside the cell but only active 6 hours outside the cell. The lifespan of HIV when it is present inside the cell 1.5 days. When it is present outside the cells only the 6 hours. The route of HIV transmission, how it is transferred? Yeah, unsafe sexual contacts, blood contaminated needles, organ transplant, blood transfusion, and a vertical transfusion from HIV infected mother to the child. Okay, mother turn the child அல்லது blood transfusion, or when we are using the contaminated blood needles, then the organ transplantation, and then in unsafe sexual practices. Okay, this HIV they cannot transmitted by the insects or by the casual contact. Casual contact when we are touching the HIV person. Through the direct contact they cannot transfer from one person to another person. Okay, through the blood, sexual contact, then the contaminated needle, organ transplant, and then in a infected mother to the child, this disease it is transferred. After getting into the body of the person, so this HIV enter inside the person, this virus enter into the macrophages. Immediately it is enter inside the macrophages. So what is the meaning of macrophage? This macrophage, what? In our WBC it is present 5 different types of WBC. That is one is known as the monocyte. Then the monocyte when it is enlarged in size they can produce the macrophages. So it is entered inside the macrophages where RNA genome of the virus replicate to the to form viral DNA. So when this HIV it is enter inside the macrophages, so that what is present, single stranded RNA only is present. That RNA they can replicate it into the DNA. They can convert it into the DNA. RNA it is converted into the DNA. So with the help of the enzyme reverse transcriptase. Reverse transcriptase என்ற enzyme the HIV virus குள்ள அந்த single stranded அதாவது single stranded 2 RNA single strand வந்து presence ஆயிருக்கு. அந்த RNA மேலே இது attach ஆகிட்டே இருக்கும். This enzyme it is making that viral RNA it is changed into the viral DNA. Can you understood? So with the help of the enzyme reverse transcriptase, this RNA genome it is changed into the viral DNA. Then this viral DNA gets incorporated into the DNA of the macrophages. அடுத்தது என்ன செய்யுது? நம்ம ஹோஸ்ட் செல்ஸில் எந்த செல்ஸில் இருக்கும்? மேக்ரோஃபேஜஸ்குள்ளே இருக்கும். ஸோ அந்த வைரல் டிஎன்ஏ they can infect the host cell DNA. Okay, then the host cell DNA directly they can produce the viral particles. So the macrophages continue to produce the virus. In this way they acts like HIV factories. So they can infect the macrophages. Automatically the macrophages DNA it is infected by the viral DNA. Then the macrophages they can multiply the number of copies of the viral DNA. Okay, so they can produce the HIV factory. Macrophages only the HIV factory. Then they can produce a lot of what? This viral DNA, HIV viral DNA. Simultaneously, HIV enters into the T helper lymphocytes. It is enter in the T cells, replicates and produce progeny viruses. So அதே சமயத்தில் அந்த HIV என்ன செய்யுது? Same time they can enter into the T helper cells. Then the T helper cells they can replicate and produce the number of viruses. The progeny viruses released into the blood attack other T cells. அப்படியே அந்த மேக்ரோஃபேஜஸ்லேருந்து வெளியில் வந்து T செல்ஸுக்குள்ளே போயிடும். T செல்ஸை போய் கில் பண்ணிவிட்டு அங்கே மல்டிப்ளை ஆகி அந்த அந்த செல்லை வந்து லைசிஸ் பண்ணிவிட்டு வெளியில் வந்து இன்னும் நியூவாக இருக்கக்கூடிய T செல்ஸுக்குள்ளே எல்லாம் போய் நிறையா டெவலப் ஆக ஆரம்பிச்சிடும். Okay, they can attack the T helper cells. This is repeated leading to a progressive decreases in the number of T helper cells. So they can go and multiply inside the T cells, then automatically reduce in number of the T cells. they can produce this viral DNA only. This T helper cells, it is infected by the what? This HIV viral DNA. Then they can enter inside the T cells, they can produce a multiple copies, they can rupture the T cells, destroy the T cells. So T cells decreases in number. Okay, so during this period, the person suffer from boosters of fever. ஸோ ஆட்டோமேட்டிக்காக இந்த சமயத்தில் என்ன ஆகுது ஃபீவர் டயரியா அண்டு வெயிட் லாஸ் இட் இஸ் அக்கர் வென் த இன்குபேஷன் ஆஃப் திஸ் ஹெச்ஐவி okay, they can incubate inside the blood, they can fight and kill the T helper cells. It is causing the fever, weight loss and diarrhea. Due to decreases in the number of T lymphocytes, a person starts suffering from infections and become immune deficient and unable to protect against any infection. அங்கே T helper cells இல்லாதனால என்ன செய்ய முடியாது, that person, infected person, they can suffer from number of other infections. They cannot able to fight and kill any other pathogens. So automatically number of diseases will affect. It is leads to the immunodeficiency. So the immunodeficiency, the T helper cells, they cannot identify the antigens, that viruses, that pathogens, they cannot able to stimulate the B cells. So the B cells, they cannot able to produce the antibodies. So number of disease causing pathogens enter inside the particular person. Can you understood, students? So other infections only it is leads the death. ஓகேவா, அந்த ஹெச்ஐவினால டெத்து கிடையாது. ஹெச்ஐவி என்ன செய்யும், அந்த ஹெல்பர் செல்ஸை ஃபுல்லாக டெஸ்ட்ராய் பண்ணிடும். அப்போ அங்கே ஹெல்பர் செல்ஸ் இல்லாதனால அந்த ஸ்டிமுலேஷன் பி செல்லுக்கு கிடைக்காது. பி செல் என்ன செய்யாது, ஆன்டிபாடியை ப்ரொடியூஸ் பண்ணாது. அப்போ அங்கே ஆன்டிபாடி ப்ரொடக்ஷன் இல்லாமல் இருக்கிறதுனால நிறைய டிசீஸ் காசிங் ஜேர்ம்ஸ் வந்து உள்ளுக்குள்ளே வந்து நிறைய டிசீஸை உருவாக்கும். they can have prolonged, that is the continuous fever, diarrhea. Due to the diarrhea and fever, body loss, weight loss, these are the other symptoms of the HIV. Due to the multiple infection, the person leads to the death. Can you understood all of you? HIV transmission, how it is causing the death of the person. The next, how we can identify this HIV infection. A simple blood test it is available that can determine whether the person has infected with HIV. ELISA test, enzyme linked immunosorbent assay, detect the presence of HIV antibodies. It is helpful to detect the ஆன்டிபாடிஸ். இப்போ கோவிட்டுக்கு பண்ணுறாங்க இல்லையா, பிளட் டெஸ்ட் ஆன்டிபாடிஸ் இருக்கான்னு பார்க்குறாங்க இல்லையா, அதே ஆன்டிபாடி இருந்துச்சுன்னா என்ன, அவங்களுக்கு கோவிட் வந்துச்சுன்னு அர்த்தம். அதே மாதிரி இங்கே எலிசா டெஸ்ட் பண்ணி பார்க்குறப்ப ஹெச்ஐவி ஆன்டிபாடிஸ் இருந்துச்சுன்னா அவங்களுக்கு அந்த ஹெச்ஐவி வைரஸ் இன்ஃபெக்ஷன் ஆகிருக்கு அப்படின்றது மீனிங். ஓகே, இட் இஸ் த ப்ரிலிமினரி டெஸ்ட். Then another one of the test, Western blot. It is more reliable and a confirmatory test. The confirmatory test is known as the Western blot test. So it detects the viral core protein. If both tests detect the presence of antibodies, the person is considered to be the HIV positive.